Hi, everyone. My name is Josh Fox Fuller. We're pleased that you all could join us for the fourth lecture in this 12-week No Neuropsychology Didactic series. From now until December, we're going to be hosting a lot of free lectures on clinical disorders, professional development, practice issues, and research skills. The No Neuropsychology Initiative was created by trainees and early career neuropsychologists to provide free, high-quality didactic opportunities. We'd like to thank our financial sponsors for the continued support of No Neuropsychology. And a couple of disclaimers before we begin. This training and the talks are not meant to replace formal education in neuropsychology, and the views of the speakers are entirely their own. Today, we'll be having a moderated questions and answers at the end of the talk. So if you have questions, please submit them via the Q&A box in the lower left side of your screen. And I'll also like to note that a recording of today's lecture will be posted within the week to our website with all the other recordings from prior lectures. Now it's my pleasure to introduce Dr. Elena Soy, who will be joining us today for her talk on computerized cognitive assessment for early detection of neurodegenerative disease. Dr. Soy was awarded her PhD degree in counseling psychology from UMass Boston. She completed her pre-doctoral internship in clinical psychology at the Tewksbury State Hospital and began her postdoctoral fellowship in neuropsychology at the University of California, San Francisco Memory and Aging Center in 2018. At UCSF, she works with Dr. Catherine Pawson on validation and implementation of novel cognitive assessment tools to support the diagnosis and monitoring of neurodegenerative disease in primary care. Dr. Soy's primary research interests include understanding racial and ethnic disparities in aging and dementia, and the early detection and diagnosis of neurodegenerative diseases in underrepresented populations. So without further ado, I'll go ahead and turn things over to Dr. Soy. Thank you for joining us today.
Thank you so much. Thank you so much for having me, and thanks for such a nice introduction, Josh. I am going to start sharing my slides now. And everything good? Can somebody give me? Okay, perfect. So thank you so much for joining this talk today. And I'm really excited to be talking to you about computerized cognitive assessment, specifically in the context of early detection of neurodegenerative diseases in older adults. This is my area of research. I'm very devoted to it. And I also think as a clinically trained neuropsychologist, this has a lot of applications across different settings that we are all working with. And so just disclosures, I don't really have any relevant conflicts of interest for this talk, and I do have funding from the Alzheimer's Association. So just in terms of introduction, I think it's very difficult for me to talk about my work without mentioning the work of my fantastic mentor. So I'll just briefly introduce her as well. I work with Dr. Catherine Pawson. She's an associate professor in the Department of Neurology at UCSF Memory and Aging Center. And she's the project lead of TabCAD, which stands for Tablet-Based Computerized Assessment Tool, which is a software platform that was designed for detection of neurodegenerative diseases in older adults. And it exists on a tablet, specifically on iPads, and we use it across different studies. And she's also the project lead of the Care Ecosystem, which is a supportive care program for persons with dementia and their caregivers. And so when I joined her lab, I was primarily driven by my interest for early detection of neurodegenerative disease, specifically using novel instruments, because based on my training, based on my clinical experience, and also exposure to the literature in the field, I recognized that there is a huge need for new tools that can be easily scalable and ready to deploy for early detection, particularly in non-specialty settings, in settings where there aren't enough neuropsychologists or neurology services. And my other big area of research is social determinants of health, and I'm specifically interested in the role of social determinants of health in late life brain health disparities. So just to start us off, I think I wanted to kind of cover our bases and talk about definitions. So according to the joint position of the ACN and NAN, computerized neuropsychological testing device is any instrument that utilizes a computer, a digital tablet, a handheld device, or any other digital interface instead of a human examiner in order to administer, score, or interpret tests of brain function. And I think this is a very broad definition, and I'm hoping that today we'll kind of be able to talk a little bit more about what are all the different modalities and what are some of the potential advantages of using a tablet-based or smartphone-based tools. So computerized cognitive testing, or CCT, is not actually a novel concept. As a matter of fact, the scientific mentions of computerized testing followed shortly after the introduction of personal computers, which happened all the way back in 1970s. And very broadly, computerized tests can be categorized into adaptations of existing traditional tests. For example, you all know and probably love the trail making test, and this now exists in a tablet-based format, where instead of using a pen, the examinee can just use their finger. And then another big category are novel tests and batteries designed specifically for computerized administration. And one of the strengths of these tests is that they really draw on all of the potential advantages that technology can bring into the testing environment. And I also wanted to briefly mention, to briefly give out an honorable mention to passive digital biomarkers, a new field that is very exciting, where people are using wearable devices or, for example, patterns of smartphone use for early detection of cognitive decline. So both computerized testing paradigms and technology use are actually on the rise, particularly in older adults. And here is figure from the paper that we recently published in Practical Neurology, where we looked at the number of PubMed publications specifically on computerized cognitive tests. And as you can see, it's rising steadily, which follows a similar pattern in the use of technology, particularly touchscreen technology, among older adults. And interestingly enough, as you can see, tablet and smartphone ownership specifically is on the rise across all the different age groups and generations. Oops, moving on. So what are some of the advantages of using computerized tests? I think the first one that probably comes to everybody's mind is precision of measurement and scoring. When we think about technology, one of the things that it can offer us is things that we as human examiners, for example, cannot measure, like reaction time, because when you measure reaction time, you have to factor in your own reaction time as the examiner. Technology does not have this drawback. We also can program our devices in a way that provides instant automated scoring and interpretation of the tests. And I think there's a little caveat here where interpretation mostly refers to just wording that is associated with a certain Z-score or a T-score, as opposed to actual clinical interpretation that still very much requires a human neuropsychologist to do. Enhanced standardized administration, I think, is a very big deal, particularly for use of technology in research studies, where you try to minimize the examiner-related variance that is introduced in the testing session, and using a tablet device or a computer, for example, really make sure that all of the stimuli and the process, that examinee is undergoing is very standardized across sessions. Another interesting thing that I think is an advantage of computerized tests is how easily we can provide different stimuli or different tests in multiple languages. So as long as you work with a multidisciplinary team of experts that can provide a culturally adapted tool, availability of these languages can actually be made very simple, as little as just the touch of a button. And then also in terms related to scoring and interpretation, computers can provide us with an opportunity to use very sophisticated or adaptive scoring algorithms. A good example of this is, for example, item response theory that has been found to be more sensitive to certain early presentations of neurodegenerative disease. And these sorts of analyses are very difficult to do when you're using paper-based tests, because that requires data input and then using statistical software to actually derive an algorithm and a conclusion, whereas a computerized device can do it all on its own. Cost effectiveness is another interesting thing that has been frequently mentioned in the literature about computerized tests, but I think it kind of is a double-edged sword, and I'll talk a little bit more about it when I talk about disadvantages. But in terms of the cost-effective aspects of computerized tests, it is definitely more cost-effective in terms of materials and supplies. They don't require use of, for example, standardized forms, of buying those huge packets of waste or CVLT tests. They also save trees, which is a very important, I think, initiative for us to think about, given the amount of paper we deal with on a daily basis. And then some of the computerized measures also provide opportunities to be self-administered or, have minimal examiner involvement in the whole administration session. And if that is the case, that provides additional advantages in terms of saving time, time both for training staff, time for administering the measure, time of the provider that the provider has to spend with the patient when they can be doing, for example, something else like record review, and also might potentially increase access, particularly to patients or participants who reside in remote areas and are not able, for example, to make the trip over to the center. However, as with everything, computerized tests also come with their certain disadvantages that are unique specifically to them. One of the important ones that I think is probably on everybody's mind are certain examinee variables. So when we work with different types of examinees, patients, participants, we have to take into account their cultural background, their experience with using technology. Digital literacy is very important, and it is inversely related to age, as well as certain disability factors. I work in a memory and aging clinic, and we see a lot of atypical dementia presentations that oftentimes present with certain motoric features that might prevent the participant to actually engage in something like tapping very quickly on a tablet. So those are all the factors that we have to consider when we are choosing our instruments. There are also technology-related issues that, again, are very unique to this certain paradigm. And these issues vary, but very broadly can be kind of put in these large categories. First is the variability in hardware and software. So for example, if I'm developing a tool that should work on iPads, how do I make sure that this same tool, when lifted and, for example, adapted to a computer screen or to a smartphone, I need to make sure that it works in exactly the same way. So measuring that variance and reporting on that variance is extremely important. And also, given how fast technology is being developed, there's a new iPhone every year, there's a new Mac every year. So I think it's very important to also be educated about different aspects of the device on which the tool is used and how that might impact performance. For example, nowadays, iPads are much more responsive to touch compared to early generation iPads. So this is the type of the variance that both as scientists and clinicians, we have to be aware of and account for if we want to use these tools for in clinical settings. Data and privacy issues are another important topic to discuss. Of course, there are data and privacy issues with paper-based tools as well, but this mostly relates to your behavior, whether you have a locked cabinet, who has access to the files. It becomes more complicated if you're using cloud services to store your data, or you're using certain services that are related to third-party servers for your tools. So those have to be compliant with the best HIPAA regulations. And also, again, as a developer or as a user, you need to be aware of what are potential privacy and data limitations. Oftentimes, when it comes to use of computerized tests, many of them use internet connection, for example, to sync the data to the server or to access certain types of features that tests can provide. Again, just an example of scoring, if you want to use a certain sophisticated algorithm, you might have to connect to the server in order to be able to do it. So that on its own means that whoever the user is will need to be aware of and probably cover certain data charges and internet access fees that might not be available in certain settings. And finally, of course, because if computerized tests are available on a digital device, no digital device is protected from certain user interface issues or bugs or tech support that needs to be provided on a continuous basis if something ever goes wrong. And then finally, if a computerized test is self-administered, that has a lot of advantages that we have discussed, but it also might come with certain disadvantages. For example, in an environment where the examiner is not required to be present altogether, we won't be able to account for certain validity indicators, things like compliance or effort. Is the person in a distractible environment? Is there anything about their performance that, for example, might indicate that they were just sitting there tapping passively on a single button? And also another huge aspect is that we'll lose a lot of qualitative data, those behavioral observations that oftentimes are critical for our decision-making when we are diagnosing or characterizing cognitive profiles of our patients. So I'm hoping that I gave you sort of a general balanced view of what are the pros and cons of using computerized cognitive testing in clinical practice and also in research. And this slide is just summarizing everything that we've talked about so far in this kind of four big categories. There is examiner-related error, examinee-related error, access, and costs. And I won't spend too much time on this just for the sake of time. So there are some important considerations that I think everybody needs to be aware of, particularly if you're considering using a computerized test in your practice or research. And those include, first, the fact that when a traditional test is adapted to a new device. For example, many of you might know that the Montreal Cognitive Assessment, which was originally a paper and pencil-based tool, now has an electronic version available on tablets, so called eMocha. However, recent publications from the Mocha developers have found that the scores on the eMocha and paper Mocha are not actually equivalent. So by translating a paper-based version of the test to a tablet-based version, you're creating a new measure. And therefore, all of the rules and all of the steps of appropriate validation are required for the new test that you have just developed. It's also important to recognize that the majority of measures available now, which have been published on in scientific literature, report substantial variability across different measures available in terms of their psychometric qualities. And so we need to pay as rigorous of attention as we do to our paper-based measures if we are to design better novel measures. So I think it's extremely important for us to look at the actual constitution of normative samples. The overwhelming majority of normative samples, both for paper-based and computerized tools, are socioeconomically advantaged, white, educated, industrial, rich, and democratic samples. And therefore, I think if we are just assuming that a tool that we have developed on a computer works really well in a certain population, we cannot just generalize it to everybody else who comes through our doors in our clinical practice. So it's extremely important to be critical when you review studies or when you review materials about who this specific instrument was validated in, and whether you think it's applicable for use in certain populations. And then finally, because computerized cognitive assessments is sort of becoming this really hot topic, and there were a lot of companies interested in developing it, there were a lot of pharma companies, for example, kind of getting interested in this space. I think it's very important for us, particularly early on in our career, to critically evaluate the marketing and the performance claims. So some of the questions you might be asking is who developed the tool and what is its intended use? There are tools out there that market themselves as they work for everybody. We work for children, work for adults, work for everybody, and yet provide very little information about all of these features about normative samples as well as validation studies. It's also extremely important to understand what are the conditions in which this certain tool has been validated. Something that has been validated in epilepsy population, for example, cannot just be lifted and automatically applied to a population with neurodegenerative disease. You also would want to consider issues like, is there continuous quality assurance and software maintenance provided by the company? If you start using this tool, particularly if you're working in the context of longitudinal clinical assessment or longitudinal research, you want your tool to be viable for as long as possible so that you can conduct your work. So it's very important to have these discussions or have this information up front about what is the longevity of the device? You know, are they going to retire it? And if they're not, how are they going to adapt it to rapidly developing technology? Again, just to give you an idea with an iPad, you know, if it worked on the original iPad, would it still work on the latest iOS? And then finally, importantly, mostly for clinicians, but also for researchers, what are the privacy and security safeguards? You know, what are the consequences of data loss or breaches that unfortunately occur across different devices, particularly computerized devices? So it's very important to keep all of these things in mind, particularly when you're considering using a tool for your clinical practice or for your research, or even if you want to learn more about the development of the tool. Those are all the things that you should be asking yourself and sort of keeping in mind. And so in terms of what's available out there, I love running the search in Google. I usually just Google computerized cognitive tests, and I've done it a year ago. And a year ago, it generated 16 million results. And I've done it yesterday, and it generated 17 million results. So as you can see, it is a very hot topic, and a lot of people are kind of involved in it and are developing a lot of tools, many of which are very promising. So I just wanted to show you some of the websites that popped up. Cogstate, all of you probably know about it. It's a great platform. CogniView, seen as Vital Signs, CanTab, and CMCI, among many, many available. And so I think now that we sort of have a general idea about what computerized tools are, I wanted to switch gears and talk about how we can specifically apply them for populations with neurodegenerative diseases, which is, again, my area of expertise and the area I work in. But before we start, I do want to also kind of make sure that we're using the same terminology. And when we talk about neurodegenerative diseases, we talk about the processes that result in abnormal aggregation of proteins in the brains. And neurodegenerative diseases are the main causes of dementia. Dementia, however, is a clinical term, and it refers to a collection of symptoms gradually impacting cognition and ability to carry out daily activities. So dementia is this umbrella term that can include many different types of syndromes, Alzheimer's disease or Alzheimer's disease dementia being the most common, but you also probably know about Lewy body dementia, vascular, frontotemporal, and other types of dementias. I think one of the important things that we need to kind of make clear in the context of this talk is that dementia is most often caused by neurodegenerative diseases, but they're not equivalent. These two words are not exactly equivalent for use. And the reason why, I think, is because there is a selective vulnerability of certain brain areas to different types of syndromes. So for example, in Alzheimer's disease, as you probably know, the most affected areas tend to be parietal areas and mesial temporal cortices. So Alzheimer's disease is caused by amyloid and tau, two specific proteins that are a part of the disease process, whereas, for example, behavioral variant frontotemporal dementia is a very different type. Even though it still causes dementia, it's a very different type of a process that occurs in the brain and is related to different types of proteins that kind of catalyze this process of neurodegeneration. And this is just an example of the different types of dementia syndromes that is being seen at our center. And as you can see, they all have sort of this very specific pattern of atrophy associated with it. And we also know that beyond atrophy patterns, that certain types of neurodegenerative diseases also affect very specific networks within the brain. So it's not just the pattern of gray matter atrophy, but also how the disease spread and along which white matter connections the pathology actually progresses. And so we know that Alzheimer's disease, for example, primarily attacks default mode network. Behavioral variant FTD is very much related to salience network degeneration. And for something like, for example, semantic variant progressive primary aphasia affects the semantic appraisal network. So the reason why I'm bringing this up is because I think it's very important for us to be precise and as precise as possible and use our knowledge in neuroscience in order to better develop and also shape our tools so that we specifically can target a certain type of presentation, as well as distinguish different types of neurodegenerative diseases through cognitive testing. So dementia in general is definitely a very significant public health priority, particularly given the fact that longevity is increasing not just in industrialized countries anymore, but everywhere in the world, particularly low and middle income countries. And so over the next decades, you can see dementia, if not addressed, will be a major a public health priority, not just from the standpoint of patient experience and family experience, but also from the standpoint of costs, given that Medicare is the primary mechanism by which a lot of older adults are covered. And I also wanted to spend a little bit of time talking about how important diversity is to dementia, not just in terms of how we conduct our studies, but also in terms of the outcomes which we try to evaluate, again, when patients present to us in our clinics. So according to US Census Bureau, over the next very few decades at this point, the majority of older adults are going to be more and more diverse and coming from many different backgrounds, not just in terms of race and ethnicity, but also in terms of their immigration status and their language status, all of which are factors that we as neuropsychologists have to be very, very aware of. And then while this demographic shift is happening, we also know that racial and ethnic minoritized populations are much more likely, they're at higher risk for incident dementia. And this is a fantastic study done by Dr. Whitmer, among others, using Kaiser data from California, where they found that African-American and American Indian and Alaska Native populations had a much higher incidence of dementia compared to non-Hispanic whites and Asian Americans. So all of this together, I think, points us in the direction of we need to be able to diagnose these diseases early, because when we think about the way in which we can address this on a public health level, the majority of interventions are most helpful when the disease is diagnosed early on. When somebody has lost the majority of their gray matter and already functionally disabled, this creates a lot of pressure, not just on the family, and a lot of suffering on the side of both the patient and the families in terms of, again, costs and planning. So early diagnosis of dementia, the majority of experts agree, is definitely a healthcare priority because it allows to identify etiological causes. Again, early on, you have more time to try and figure out which specific protein is causing this disease through longitudinal monitoring, as opposed to when the patient is presenting later on in the disease and the syndrome is just so complicated because there's so many things that are affected. It also allows to inform and coordinate medical care, specifically thinking about multidisciplinary care when you can really utilize all of the resources that the patient might need, whether it's occupational therapy, physical therapy, nursing, social work. You can work with many different providers and coordinate care better. It also enables planning for the future for the families. It allows them to think about their financial futures. It allows them to consider the cost that they may accrue over time. So it just gives them a lot of agency to work with their doctors when they're faced with this horrible diagnosis. It also allows to address potential safety issues. Again, this is all about thinking ahead. We probably are all aware that there are major safety-related issues in individuals with Alzheimer's disease who, for example, might forget to turn the stove off or wander off in the neighborhood and get lost. So all of these things are preventable as long as we, again, diagnose early and make plans. It allows to connect families with interventions. This is, again, very much a care pathway where families might be able to benefit from certain caregiver intervention, preparing caregivers on the types of activities that they can engage in to take care of themselves and of their patient. And then finally, identify appropriate candidates for clinical trials of potentially disease-modifying therapies. And I will not mention aducanumab because it's a very controversial topic and I don't want to spend too much time talking about it. But I do think that one thing that we've learned from the aducanumab experience is that the disease-modifying treatments, when they become available, will be most helpful to older adults who are diagnosed early on. So early diagnosis becomes a key priority for us. If we are to change the overall impact of this disease, we need to be able to diagnose and detect early. Unfortunately, most of the previous work, including our work presented here, shows that there are significant disparities and inequalities in terms of early diagnosis. In a recent study that we published in JAMA Neurology, we looked at 10,472 California Medicare beneficiaries who had an incident diagnosis of either MCI or dementia. And MCI, which also stands for mild cognitive impairment, is frequently perceived as an early prodromal stage of dementia and is associated to progression to dementia over time. So we looked at whether racial and ethnic minoritized populations were less likely to receive an incident diagnosis of mild cognitive impairment compared to dementia. So that was our way to look at the timeliness. How timely was the diagnosis? And we indeed found that Hispanic, Black, and Asian beneficiaries were all much less likely to receive an early diagnosis of MCI compared to dementia. And this is not just an issue that we see here in California or even here in the States. The World Alzheimer Report published by the Alzheimer's Disease International, it was published just last month, and we submitted an essay there talking about that there are significant disparities in early diagnosis everywhere in the world, and specifically in low to middle income countries. As a matter of fact, the rate of underdiagnosis or late diagnosis is significantly correlated with the income of the country. It's inversely correlated to it, as you can imagine. So the rates of underdiagnosis in high income countries is about 60%, whereas the rate of underdiagnosis in low to middle income countries is roughly more than 90%. And within high income countries, countries like Western Europe and Northern American countries, racially and ethnically diverse older adults are at much higher risk of underdiagnosis. And this has been found in studies from Denmark, Norway, United Kingdom, and the United States. So I think these findings highlight how when there is this huge need for early diagnosis of this very life-changing disease, racial and ethnic minoritized populations are unfortunately much less likely to get access to those services. And so this is where I think neuropsychologists come in, and as neuropsychologists, one of the roles that we play is characterizing cognitive profiles and cognitive impairment in older adults. For those of us who work in memory clinics, that is our primary role. So what are some of the issues that we are facing? First of all, I think, and I hope the majority of you will agree, that the majority of the measures that we have, the tools that we have, were published, developed, and validated in Western countries. Most of the samples, again, were highly socioeconomically advantaged non-Hispanic white participants. The number of neuropsychologists who have competency to work with different ethnically and racially diverse groups, or even possess proficiency in non-English languages, remains critically low. It remains critically low, again, not just in Westernized countries, but also in countries where there simply is no training in neuropsychology, and yet the need there might be even higher than here. And then finally, the majority of the measures that we use are significantly influenced by linguistic factors, educational factors, and not just years of education, as fantastic Jennifer Manley showed. Quality of education plays a huge role, and this is something we very rarely take into account. There are also socioeconomic variables that impact cognitive performance, as well as cultural factors. So all of those things critically limit neuropsychology, and I think our ability to truly participate and facilitate early diagnosis. And then in order to kind of, this is a topic of great interest for me, particularly barriers to early diagnosis and barriers to care. So we did a qualitative study with 18 dementia experts around the world from countries that had the greatest projected growth of older adults in the next 30 years. And the countries are presented here. And we found some very critical key limitations, as well as the needs for neuropsychology related services in those countries. First of all, the majority of our participants said that access to neuropsychology is extremely limited, and cognitive measures, for example, mini-mental state examination or Montreal Cognitive Assessment, are frequently administered by medical specialists, people who do not actually have specialty neuropsych training. Current measures were also found to be too long for wide-scale applications. Very few countries had the ability to have tertiary neurology centers where you would have an hour or a couple of hours to examine patients. They usually have 15 minutes to collect everything: history, labs, imaging, and cognitive testing if there is time for it. Current measures were also either poorly or not at all validated in their populations. So oftentimes our participants reported having to use, for example, an English MOCA with a translator trying to adapt it to the local language, or using any sort of tools that they had at their disposal if they didn't have a translator, like using a Google Translate, for instance. There was also poor applicability of measures due to language education and literacy factors. Again, there are lots of countries out there, and there are lots of populations out there that remain very low literate. And I think the majority of the measures that we use require at least certain level of reading and writing ability in order to be able to perform on them, or even understand what the test is requiring you to do. There was also a report of poor applicability due to cultural biases. For example, notorious for this now is Boston naming test, but also certain naming items, for example, on the MOCA, might not be available to certain populations. For populations that have never seen a rhinoceros in their life, that might not be something that would be culturally available to them. And then finally, the overwhelming majority of our responders said that most of the measures available had very limited diagnostic accuracy, as well as sensitivity to severity. That again gets to this early diagnosis, early accurate diagnosis problem. So how can computerized tests help with these issues that we have discussed? And I'm just trying to check the time to make sure. How much time should I leave for questions? I probably have maybe 10 more minutes.
Yeah, I would say maybe 10 more minutes, and then 10 minutes for Q&A. Sounds good. Yeah.
Fantastic. All right. So this was the big question for me when I started doing my research, because this is exactly what I wanted to do. Being aware of these issues, how do we develop tools and how do we implement them in a way that can help us at least move the needle a little bit in the right direction? So I think knowing that access and use of technology among diverse persons is growing globally. For example, smartphone use is actually on the rise, even among individuals who have low literacy. And it would also allow us to harmonize and standardize data and algorithms across different nations. I think, when I think about when I was starting doing my research in my doctoral program, and I had very limited access to any samples, and I was sort of like scrambling for data and trying to find what are the answers that I can generate from a certain data set that existed before me. I think now we live in the era when global harmonization of data is definitely possible, and it just requires coordination and facilitation of different types of stakeholders around the world, which I'm hoping to show you in a little bit. It also allows, computerized tests would allow for deep and wide data collection, meaning that we can both collect a lot of information about a single person, and also in wide collection means that we will be able to do this hopefully in a time and cost-effective manner across very different individuals. And then, as mentioned before, we can implement sophisticated normative algorithms that would not require examiner scoring, thus again saving time. And then finally, computerized tools, at least the types that I have worked with personally, usually involve less costly development and cultural validation of appropriate measures. So now I want to talk a little bit about the UCSF Brain Health Assessment, which is the battery that I work with most closely, and I'm hoping you guys will get to know about, and if you have any questions about it, feel free to reach out. So the brain health assessment, or the BHA, is a very brief 10-minute iPad-based battery designed for efficient detection of neurocognitive disorders, specifically in non-specialty settings. And it features easy and quick administration. We do have an examiner administering this test right now. It has an attractive user experience and also has automated scoring and provider feedback. So here are the components of the brain health assessment. We have a task of called match, which is based on digit symbol paradigm. It measures executive functioning and speed. We have an associative memory task where persons are asked to remember a face and their favorite things and are later asked to recall them. Line orientation, which is an adaptive measure of visuospatial function. And so what's cool about this measure is that the better the person is doing, the harder the test becomes. And then the worse they're doing, the easier it becomes, actually. Animal fluency is an optional task, and it's administered in exactly the same way as you would. How many animals you can name in a minute. And then we also have some optional informant surveys that I won't get into too much today. So few of the things that I think make BHA a very helpful tool for early detection is the way that we have approached developing norms and composite scores for this specific tool. First, we do have nonlinear regression-based norms that correct for age, education, sex, and language of administration simultaneously. And these norms allow for greater accuracy of prediction because, as you know, regression-based norms are highly individualized, therefore allowing you to basically adjust for all of the demographic factors at once, as opposed to using box norming that provide much more variance and error around the Z-score. And then in addition to each of these regression-based Z-scores that we have, we also have a composite score called a cognitive score, which is based on performance on match, favorites, and animal fluency, and kind of captures the overall global cognition composite. So the BHA was validated originally at the Memory and Aging Center in our cohort of typical and atypical dementias. By typical dementia, I mean dementia of Alzheimer's type, and atypical, everything that is not Alzheimer's type, or rather non-amnestic Alzheimer's type. So, and we found that the BHA significantly outperformed the MoCA in terms of detecting impairment early at the mild cognitive impairment level in both individuals who likely had Alzheimer's type pathology and also non-Alzheimer's type pathology. And also, I think another important validation piece that we've done is we also looked at neuroanatomical validity of each of our measures. So remember how we looked at selective vulnerability? One of the things that I think we can derive from neuroscience and really use in our studies is making sure that the measures that we have translate into the similar brain centers that we would like to assess as a part of our evaluation. So here, for example, we're showing that our memory test favorites really localizes to gray matter volumes in medial temporal lobes, which is exactly where we would want them. Match localizes to frontoparietal and subcortical areas, which largely underlie executive functions in humans. And line orientation and visuospatial measure is unsurprisingly very much related to right parieto-occipital areas, which basically is a part of the dorsal visual stream. So after we've done our validation in English speakers, we also wanted to make sure that the tasks that we're using can be used and it can be equally as helpful in individuals who speak Spanish. And so we validated the BHA against the MoCA. Again, it significantly outperformed the MoCA in the US-based Latino sample, mostly of individuals who immigrated to the US from Latin American countries. And we also published a study that was based in Cuba in a Spanish-speaking Cuban sample, where again, we found that the BHA measures significantly outperformed the MoCA in terms of early detection. One other piece of work that I think is very important for us to consider when we think about early detection is how reliable the measure is. You know, beyond what we can do kind of in a single, at a single time point, we also want to make sure that if there is a need for longitudinal follow-up, that we will be able to reliably detect change over time. And the BHA composite actually showed excellent longitudinal reliability at 1 year. So the R coefficient was 0.86. And then we also saw that based on reliable change indices, the majority of the individuals who were controls and remain controls actually were very stable, whereas 43% of individuals with MCI have declined and 71% of dementia patients have reliably declined, which again just shows that the tool is able to track cognition reliably over time. And then this I will skip through, but we also looked at the BHA performance at home and in the examiner office, and we found that it is a very stable tool, both in examiner administered and self-administered settings. And this is something that is in development right now, so I won't spend too much time, but we are developing a self-administered version of the tool that can be easily deployed again in settings where the examiner might not be available. And then finally, one of the things that I wanted to highlight in relation to etiological or accurate diagnosis is that we live in the era of biomarkers when, for example, we can use the tools and technologies available to us to now fairly well predict whether somebody will have Alzheimer's type pathology, because Alzheimer's disease, as I mentioned, is defined by abnormal accumulation of amyloid and tau proteins in the brain. And the positron emission tomography or PET studies actually allow for in vivo detection of pathological changes associated with Alzheimer's disease specifically. And again, to highlight what it means for treatment and patient care, if there are potential disease-modifying drugs on the horizon, then etiological diagnosis will be critical, because the majority of the companies that develop the drugs don't develop drugs for dementia. They develop drugs for very specific types of proteins that cause dementia. And so it's very important for us to be able to make etiologically informed diagnosis in order to treat our patients right. And so in our studies, we found that the brain health assessment was sensitive to amyloid beta positivity on PET in older adults with cognitive impairment. And we found this both in individuals who had very mild impairment and also individuals with dementia. And in addition to amyloid, another protein that causes a lot of issues in Alzheimer's disease is, of course, phosphorylated tau. And so we did a PET-based study in individuals who had both tau PET and took our tool, and found that the performance on certain types of tasks that measure certain types of skills were correlated with tau uptake in the areas where we think this function lies. For example, our associative memory task was very much related to the amount of tau protein that was seen on PET signal in the mesial temporal lobes. Match, which is a task of executive function and speed, was again associated with frontoparietal tau uptake. And finally, our visuospatial task line orientation was very much related to tau signal in occipital-parietal cortices, which, as you all know, underlie visuospatial functions. We also looked at the sensitivity of our tool. If we were to go beyond clinical populations and start studying older adults who don't yet have cognitive impairment, but are amyloid positive on PET, which a lot of researchers are currently calling preclinical Alzheimer's disease. So they already have amyloid in their brain. So if we follow them longitudinally, are we able to then predict that they are more likely to have cognitive decline? And we indeed found that in older adults who were followed over 5 years and remained stable on paper and based tools, we found that the BHA composite was actually declining much more significantly in those who were amyloid positive, as opposed to those who were amyloid negative. And we did not find anything like that using the MoCA. All right, so just to summarize everything, how is this helping? I think we're trying to address this issue of early detection through sensitive norming, through addressing reliability, through taking validity studies, not just for syndromic validity, but neuroanatomical and biomarker sensitivity validity studies, as well as reducing errors related to scoring and prediction, and developing tools that can be used by non-neuropsychologists if they're given appropriate training and understanding of the results. So this is one of the slides that I really wanted to show. I am actively involved with the Global Brain Health Institute, which is an initiative that exists at UCSF and Trinity College Dublin, where every year we train a group of amazing, fantastic fellows from everywhere in the world who study brain health and particularly brain health equity. And so we have collaborated with numerous fellows around the world, and all of whom have been using our tool, the BHA, to validate, to implement, to detect cognitive impairment in primary care. And I think beyond just having this very rich research data, one of the most important contributions that all of these amazing individuals made is they helped us culturally validate the BHA. You know, we worked with focus groups, we worked with medical anthropologists and local partners to make sure that whatever we put into our stimuli or into our instructions would make sense for the population for which BHA is being used. So we have very culturally distinct versions of the tool across different countries and nations. And so these are just some examples of our partners. We overall have more than 400 users across 65 studies that we support, and we're also actively using the BHA in primary care here at UCSF. So I wanted to end with some future directions for the field. I apologize for going so fast in the last few minutes, but I wanted to make sure we have time for questions. But I think as a field, we need to think about developing and culturally validating appropriate measures and norms. And this involves collaborations with individuals outside of our disciplines, anthropologists, language specialists, medical specialists, and local communities, more specifically in test development. And I also think we need to get out of this mindset of sort of standard norm development, you know, the tests that have been developed and published in 1940s, you know, and using the same pipelines to develop tests nowadays. I really think we have to utilize the best scientific advancements and technologies in order to make our measures better and therefore serve our patient populations better. I also think it's critical for those working in the field of computerized cognitive testing to systematically assess examinee-related variability. We always collect data on digital literacy and also technology use. And having that be a standard part of the assessment, I also think is critically important because that might impact the interpretation of the results. Another huge part that I'm hoping to make a call for among early career neuropsychologists and established neuropsychologists is to support and promote international collaborations. Really, we learn so much from each other, and living in the world where we're so connected through the power of internet, I think just makes these collaborations exceptionally valuable for all of us. And then finally, we won't be able to do any of this, I think, if we don't train, hire, and promote diverse neuropsychologists that come from many different backgrounds, socioeconomically, linguistically, culturally. The only way I think to move the field forward is to have people have power to make the decisions about who gets hired on and who gets promoted and who gets to do research. So this is a critical time in the field for us. And I think it's extremely important when you work as a part of the community to be aware of who the community is. So I'm going to end on this note. And I just want to thank everybody, particularly my fantastic team, all of my UCSF and GBHI collaborators. This slide could have been 10 slides. So I'm just going to stop there and tell you that if you ever have any questions, I'm more than happy to answer them via email. I am on Twitter, but I'm really bad at it. So if you really want to get a hold of me, please email me. And I'll stop right there.
Awesome. Thank you so much, Dr. Soy, for this wonderful talk. I am a little biased. I was furiously scribbling notes down because one of my dissertation studies is actually validating a computerized cognitive test. So hopefully other people in the audience are in the same boat. So we have a couple of questions here, which we'll just kind of go through. I wish we had more time, truly, but it's, I think everything you said in this concluding point was really helpful to hear. One, I think a couple slides back, you actually mentioned, or there were some of those international collaborations. They were for not just Alzheimer's disease, right? I think there was one of frontotemporal dementia, one of Parkinson's disease.
Yep. Yeah, let me.
Yeah, you can go back if you want to. This question, it sort of speaks a little bit. I'm looking over here at my other slide. It speaks to this, I guess, this question of, is it, in your opinion, is it appropriate to use computerized assessments for all different forms of dementia? And in particular, this person saying how it could be difficult for someone with, say, severe behavioral impairments in BVFTD or if they had severe tremor and Parkinson's disease. So what are your thoughts on that?
Yeah, I think it's an interesting question. I'm wondering, whenever I get these questions, I always think, well, what would I do as a classic neuropsychologist, right? Like I will come in with my paper and pencil test, ask them to copy a ray figure, right? And try to make sense of it. So I think computerized cognitive tests are not necessarily that different from paper and pencil measures in this regard. So I think there's always room for, again, a human neuropsychologist to be there, interpret the results, which usually happens in very specialized clinics that have neuropsychologists on staff. But I also think there is an important consideration, for example, for individuals with behavioral variant frontotemporal dementia that we see a lot here at UCSF. They are actually most of the time able to follow instructions if you're sitting there with them. So an examiner kind of moderated session is usually works really well. And I think for individuals with Parkinson's disease, there is an aspect of motor impairment, but we also know that specifically in Parkinson's disease, the degree of their motor impairment is actually correlated with the degree of their cognitive impairment. So, if you're using something like a UPDRS scale, then it will be correlated with global cognitive functioning. So I think that variance is important to consider, and using your clinical judgment as to whether, a certain score is impacted by motor impairment, you would do this in exactly the same way as you would when you're interpreting somebody's ray copy. Nice. I hope that answers that question.
Yeah, I think so. I mean, I think I kind of agree with that, but there's always a space, right? I mean, whenever there's always a space, we just kind of have to be creative, like you said. And then I guess another question that we had was, in terms of this norming process, right? You spoke to how we have to be innovative and creative as we, launch into these future directions of the field, which I completely agree with. So in terms of maybe the studies you've run or studies you're familiar with, how do you go about, do you use like a standardized computer to norm it, or is it sort of a bring your own device thing, and how do you adjust for those variances in screen size and screen brightness, or what sort of methodological thoughts do you have?
Yes, fantastic question. And this is something that we think about every day. And I think we err on the side of caution because we run so many research studies where a standardized protocol is so critical. So we use exactly same devices. We use only iPads. That's why our tests are only available on iPads. And we work closely actually with our developers on making sure that every new iteration of iOS that comes out is tested against our existing paradigm to make sure that the reaction times are the same. That if a person makes an error, that it's basically recorded in exactly the same way. And so I think for individuals who use different types of devices, there is definitely room for wide scale, right? If you can allow to have noise in your data, if, for example, you're not trying to make individual level predictions, you're not diagnosing a single individual patient, but rather, for example, studying how patterns of smartphone use are related to, safety concerns, you can do this on a group-based level, I think, using many different devices and then just control for them in statistical analyses. But I think if you are trying to use a tool to make individual level predictions, having a standardized measure is critical because, it would be the same as using like an A4 versus an A5 version of trails. And that is just creating a lot of variance that you would want to get rid of as you're doing a clinical assessment.
That makes a lot of sense for sure. It's something that I think about a lot for my own study.
Absolutely. And I think it's very important that people like you are thinking about it because you might come up with something brilliant and we won't have this problem in the future.
We'll see. I don't know if I go that far. Then just one last question, since we're at the top of the hour and I don't want to hold you too much longer. So for someone like yourself, it's clear you've been a trailblazer and, kind of connecting all these international partnerships together. Definitely, it sounds like the GBHI is a big thing you'd probably recommend, but what other advice might you have to trainees or early stage people for kind of fostering these international collaborations?
Yeah, I think that's a fantastic question. And this is something that I also have learned to do as, I'm an international neuropsychologist myself. And so I think my most important advice, if I could give one, is don't be afraid to reach out. Scientific community and I think neuropsychology community is very tightly knit. We're a very small group. It might seem like there are a lot of people, but they're actually very few. So I think reaching out, if you come across a paper that really fascinates you and you would really like to learn more and maybe contribute to somebody's work, I get these emails all the time and I'm more than happy to respond to them. And I think one of the ways in which we can connect to each other is actually using each other's work as inspiration. So if you have somebody who, again, you're very interested in working in, or just want to learn about what they're doing, go to their lab meetings, please do not hesitate and reach out to them. I would say, I cannot actually put a number on it. It will be with huge confidence intervals, but 80% of the time, people will respond and will be more than welcome to have you in there as a part of their teams. So I think when you think about international collaborations in particular, it's very important to consider what are the different healthcare settings within which people are working. This is one of the things that I am still learning, having not been involved in healthcare systems across all the different countries. But I do think this is a huge part if you want to do health-related research to do on your own, research as much as you can about what the different healthcare systems, do they have a national dementia prevention plan, and do they have a treatment plan? And this will really put you above and beyond people who are probably reaching out to these people anyway. So do your research, don't afraid to reach out. And I think more than anything, be inspired by somebody else's work. This is the thing that keeps us going. And I think for international research in particular, now is the time. Now is the time to get involved. Now is the time to make those connections, to make neuropsychology a brand new international science.
That's awesome. I think that's really helpful to hear that as a recommendation. So I'm sure I could ask you questions for a long time, but we'll stop here. Just as a reminder for people for next week, we're going to have Dr. Michael Parsons from Massachusetts General Hospital talk about central nervous tumors at 3 p.m. Eastern on the 11th. And thank you again, Dr. Soy, so much for coming today. We really appreciate you taking some time to talk to us.
Thank you so much for having me. It was such a pleasure. And again, feel free to reach out. I'm always available and I usually always answer my emails. Maybe not on time, but I do.
All right. Thank you so much, Dr. Soy.
Thanks,
have a good
everybody.
rest of your evening. Bye, everyone.
Bye.